Hello everyone, I'm Saeid from Dr. Caswell Lab at Department of Pathobiology at OVC, and today I'm going to talk about my research project on bovine respiratory disease, which is an important issue for the cattle industry. Beef cattle, like any other animal, has defense mechanism to protect them against illnesses. Before, we were only thinking that stress and viruses make their immune system weak, and that's why pneumonia happens. But some of the researchers are suggesting the cattle body itself can also be responsible for the lung damages, and this is because stress and viruses can change the programming of the cattle immune system in a harmful way. To know why some calves get sick and why others not, and make good treatment, we need to understand how calf body react to stress and harmful microorganisms. So in our work, we study how different transition period stresses such as weaning and transport affect lung health. To see if their combination is related to bad health, we use high-risk auction calf, which go through various stresses and may have higher loads of harmful organisms because they are coming from different farms and they commingle in the auction barn. To answer our questions, we have designed several studies and we compared the initial lung health markers between calves from Elora that are either abruptly weaned and suffer from weaning stress with less stress to stage weaned calves. Also, we have calves which are abruptly weaned and also transported, and for our experiment last fall, we got them from a new Lescard research station. The other thing that we check is how these changes in the lung are later help the bacteria and viruses to cause tissue damage. For that, we spray calves having different level of stress with killed pathogens because these sprays produce inflammation similar to natural infection. The good thing about this method is we don't need to expose calves with live harmful bacteria and viruses, and so we can work on many animals under the normal barn condition. In our studies at Elora Beef Research Center, we washed the lungs of calves with an sterile solution to measure the markers of lung health in the lab. We take blood samples to check how inflammation happens at the body level. We test for a hormone related to stress in the fecal samples to see how stressed these calves are. For future, we are interested in understanding the role of genetics in pneumonia, so we also sample hair for those studies. Here are some of our early findings. As you can see, the level of inflammation markers in the lung washes is related to the level of stress and was higher in medium risk calves from New Lescard research station, and these calves had been both transported and abruptly weaned comparing to the other group. We saw a similar pattern in inflammatory markers of blood, and again, the higher the level of stress, the higher were the number of white blood cells which can react to pathogen and cause inflammation. So far, our preliminary findings are supporting the hypothesis that stress because of transport and weaning encourage inflammation. And based on a previous work in our lab, this may make at-risk calves susceptible if they get harmful BRD pathogens. So we are planning to explore these phenomena in future in the high-risk auction calves. And we are hoping with this research, we can help farmers to reduce their losses because of the bovine respiratory disease, and also help them to reduce the consumption of antibiotics by developing new treatments for bovine respiratory disease. And this way we can have a healthier, better product for consumers too. And finally, I would like to thank our funding agency, because without their help and support, this project was not possible, especially OMAFRO and beef farmers of Ontario. And Thanks everyone for your attention.